Hello, chemists. Michelle Glass here. And congratulations. Oh my goodness, you made it to the last of our chapter 2 videos. I know you felt like, I'm never going to get to the end of this chapter. I felt that way too. Notice I had like multiple wardrobe changes and I had a cold and I lost my cold in the making of these videos. So thank you so much for sticking with me. This is our final video. We are finishing up our organic macromolecules with our fourth, which is the nucleic acid. Remember, you're paying attention to the structure, examples, and functions of nucleic acids. Let's begin with the monomer. The monomer for the nucleic acid is called the nucleotide. And a nucleotide consists of a phosphate group, and then we can pair with it a 5-carbon sugar. I'm drawing mine, looks like a house, right? So each one of those points is a carbon. So I have 5 points, and that's my sugar. And then we have what's called a nitrogenous base. That's paired here. And actually, I need to adjust my drawing so that my phosphate is kind of sticking up in the air. Kind of like this. Something along those lines. Okay, not perfect. Now, when we talk about the nitrogenous base, you can have, I'm gonna list it right underneath. You can have adenine, you can have thymine, you can have guanine, you can have cytosine, and you can have uracil. And so I'm going to actually change how I've drawn this. So then I'm going to have, beside my adenine, I'm going to have either thymine or uracil. Now this, rewriting it and drawing it together is important because we do have what's described as complementary base pairing. So adenine will pair with thymine, or adenine will pair with uracil. And the difference there has to do with whether we're talking about a DNA molecule or an RNA molecule. So we'll see some of those differences as we progress in the future. So one easy memory device is that the apples grow on trees. So that's how you can remember that adenine pairs with thymine. Or you can remember the car is in the garage. So in that case, it's the cytosine that pairs with the guanine. And very often we just will refer to these nitrogenous bases by the first letter. So we'll talk about A pairs with T, G pairs with C. And then we'll see that uracil is found in our RNA molecule only. So we'll talk about uracil when we're talking about RNA. Let's go ahead and list out. We have 3 different examples. So we have DNA, which stands for deoxyribose nucleic acid. So deoxyribose, OSE ending, remember, tells us sugar. So this is telling us what type of sugar we'll see for the 5-carbon sugar of our DNA molecule. We also have RNA, and one of the key differences here is that the R is standing for ribose. Again, OSE ending tells us sugar, and so ribose is the 5-carbon sugar in the RNA molecule. And then we have a molecule that we've actually mentioned before, which is ATP. And ATP, remember, stands for adenosine. So that is like a combined word that tells us adenine for the nitrogenous base. And then the adenosine is telling us about the sugar. So it's kind of like a combined word to say like it's adenine and a 5-carbon sugar. And then triphosphate. So one of the differences that we'll see in the ATP molecule is it doesn't have just the 1 phosphate group, it has 3 phosphate groups. Okay, let's talk a little bit more about structure. Okay, so I redrew my nucleotide there to give myself a little bit more room. When we talk about the DNA structure, that's kind of this drawing over here, where you have a double-stranded molecule, and it's the bases, so like the adenine and the thymine, or the guanine and the cytosine, that are being paired together in the middle. So that's where we talk about that complementary base pairing. So you can imagine, like, this is classically described as a ladder, right? So the sides of the ladder would be that sugar phosphate backbone. And then the parts of the ladder that you stand on, the rungs of the ladder, would be those nitrogenous bases, hydrogen bonded together. So the hydrogen bond between the adenine and the thymine, or between the cytosine and the guanine. So when we talk about DNA compared to RNA, we see that DNA is double-stranded. We see that DNA has deoxyribose. And we see that DNA has adenine paired with thymine, guanine paired with cytosine. And remember, deoxyribose. When we talk about the RNA molecule, it's going to be single-stranded. So we can draw RNA kind of like a ribbon, where again, the backbone is the sugar phosphate. And remember, the sugar here is ribose. And then we have the nitrogenous bases kind of coming off of the ribbon. And we're going to have adenine, uracil, guanine, and cytosine. So when we talk about uracil, we see that that is part of the RNA structure, not the DNA structure. So again, comparing structure for DNA and RNA, DNA is double-stranded, has the sugar deoxyribose, and has adenine, thymine, guanine, and cytosine. RNA is single-stranded, has the sugar ribose, and has the nitrogenous bases adenine, uracil, guanine, and cytosine. To finish up, let's look at our ATP. And here what we can do is kind of draw our adenosine maybe as like a box, and then draw your 3 phosphate groups. And you can just kind of represent those if you want, like as a P with a circle around to represent those phosphate groups. And here's the thing that's important. Each one of those phosphate groups have a negative charge. So you've basically jammed together negative charges. And we know already that opposite charges attract and like charges repel. So you're like holding together really close in proximity these negatively charged phosphate groups. And so this creates like what we call a high energy bond. And so we have 2. The highest energy is that one that's more on the outside. So between like the second and the third phosphate, that's your highest energy bond. And then between your first and your second phosphate, that's another high energy bond. And so think about this as like if you have that spring and you're holding it together, and the minute that you release it, you get that energy from that spring's release. We have the same kind of idea here. So the holding together of these negatively charged phosphate groups creates this high energy bond. And the minute that phosphate can like get away, you get this big release of energy as it's just like whoosh, leaves that bond, right? So this is our energy. So very often when we talk about this, we'll see a DP. Now the D stands for diphosphate as opposed to triphosphate, which would be 3. So ADP is an adenosine di, as in 2 phosphate, plus a phosphate group. To make this takes energy. So we're going to, in our reactants on the left side of our equation, we're going to write ADP plus P, phosphate, plus energy, gives us our ATP molecule. So our ATP is our product here. And this would be an example of an endergonic reaction. This is a reaction that requires energy. When we break down our ATP, so now let's put our ATP on the left side of the arrow. Let's make it be the reactant. What we see happening is when we break down our ATP, we get that ADP back out, the adenosine diphosphate back out. We get that phosphate group released, and we get energy given off. So when we break down ATP, we get energy released. This is kinetic energy. This is energy that can be used for work in the cell. This is an example of an exergonic reaction. So one of our key functions of nucleic acids is this ATP molecule is our energy molecule. When we talk about the DNA and the RNA molecule, we talk about these as the hereditary molecules. They're going to code for protein synthesis. Synthesis means to make. We can also say that these are molecules involved in directing protein synthesis. So they're like in the kitchen helping to stir up and make that protein. I mean, that's kind of a silly analogy, but you have this idea. Okay, that is it for chapter 2. Yay! As always, submit your questions and take care of yourselves and each other.